In this tutorial, we are going to talk about how we can use the modeler to model the missing amino acid residues in the protein data bank file. For this tutorial, we are going to use 1QG8 as an example. This is native magnesium coating SPSA from Bacillus subtilis. As you can see it here that this protein structure is a high resolution structure having a resolution of 1.5 Armstrong. And this structure is determined by using X-ray diffraction analysis technique. To get an idea that how many amino acids are missing in this three-dimensional structure, let's download the protein data bank file. We hope so that you will have a very good idea how to download the PDB file. To download the PDB file, just click here. There will be a drop-down menu, and in this drop-down menu, click here on this PDB format. Once the protein structure is downloaded from the protein data bank, then right-click on it and then open it either with the notepad or the WordPad. When you will open the structure into the WordPad, then you will have this description. In this description, you can find out that how many amino acid residues are missing in the protein data bank file and what is their location. To get an idea about the missing residues, press Ctrl F. Afterward, write down missing and then click Find Next. When you will click on the Find Next, then if they will be the missing residues, it will be highlighted in front of you. As you can see it here, that these are the missing amino acid residues into the protein data bank file. We are going to have an idea about the type of the amino acid residue, its chain, and its location. For instance, here we are going to have an idea that the first amino acid residue which is missing, that is glutamate. This belongs to chain A and its position is 134. The second amino acid residue is asparagine. It also belongs to the chain A and its position is 135. And the third amino acid is arginine. It also belongs to chain A and its position is 136. Similarly, we are also having an idea that there is some amino acid from position 218 to 231 which are also missing into the protein data bank file. We cannot use this protein data bank file for the docking studies or the molecular dynamics simulations until unless we are not going to fill these gaps. To fill these gaps, we are going to use the modeler. So let's move to the modeler and let's try to see how we can use it. To use the modeler, we need to have three scripts: script 1, script 2, and script 3. We hope so that you will be familiar with the modeler and you have already practiced the homology modeling using the modeler in the previous tutorials. All of these scripts are the Python scripts, and we will not recommend you to change anything in these scripts. Otherwise, it may create a lot of mess for you. To use these scripts properly, we are going to rename our protein data bank file from its original name, which is 1GQ8, to QPDB. So let's rename it. You can also use the original PDB name, but for this you need to change something in the scripts, and we wouldn't recommend this for the beginners. If you are expert, then you can do it. The second thing which we need to use these scripts properly is the full-length amino acid sequence. We have already this full-length amino acid sequence here. Now you can ask a question from where we can get the full-length amino acid sequence of our protein. The answer is pretty simple. You can get the full-length sequence of the protein from the protein data bank. How? This time, click on this download files. There will be a drop-down menu. But this time, you should not click on the PDB format. This time, click on this FASTA sequence. When you will click on this FASTA sequence, then the full-length protein sequence in the FASTA file will be downloaded in your system. Now question comes here, how we can use these scripts? We have placed all of these scripts into one folder, and name of that folder is repair1. Let's copy this folder and paste it into the modeler folder. Where our modeler folder is present, our modeler folder should be present in the C drive, and normally it is installed either in a program files or program files x86. In our case, the modeler is present in program files, and here there is a modeler 10.1. We will open the modeler 10.1 and then we will move to the bin, and inside the bin we will paste our folder, which is repair 1. After placing the folder in the bin, we will go to the start menu, and in the start menu we will type the modeler. As we will type the modeler, there will be this black color insignia. Right click on it and then click on Run as Administrator. This black color window will appear in front of you. Now in this black color window, type cd bin. What does it mean? It means that we are requesting our system that please move to the bin. As we will hit the bin, you can see it here that right now we are in the bin folder. Now let's move to the repair 1 folder where our scripts are present. To move to the repair folder, type cd and then repair 1. After typing this, let's hit the enter button. And as you will hit the enter button, you can see it here that right now we are in a repair folder. To check the content of the repair folder, type dir. As you will type the dir, all of the files which are present in the folder, they will appear in front of you. Now this is the time to run the scripts. The question comes, how to run the scripts? Super easy. Type mod 10.1 space script 1.py. Please be aware of the spell mistakes. Any spelling mistake can create a mess for you. After writing down mod 10.1 space script 1.py, hit the enter button. As you will hit the enter button, then the script will be executed and the job will be done. Now if you will go back to the folder, then you can see some new files will appear in front of you. And one of the very important file which will appear in front of you is this one, that is PDB. And if you look at the extension of this file, then this is seq file. What does it mean? It means that this file will contain the sequence which is basically obtained from the qpdb file. Now remember what the script 1 did. Now please let us tell you that the script 1 basically extracted the amino acid sequence from the protein data bank file. Now after successfully executing the script 1, this is time to execute the script 2. Before executing the script 2, we need another file, and that file is an alignment file. The question comes here that how we will get the alignment file. To get the alignment file, we need to first align the sequence which is extracted from PDB file and the full-length protein sequence which is downloaded from the protein data bank. Now to align the two sequences, we will use the pairwise sequence alignment. And obviously, we will align them by using the global alignment. We have talked about the global alignment in detail in our previous course, which is Learn Bioinformatics from Scratch. To pairwise align the sequences, we are going to use the emboss needles tool. We will copy and paste the two sequences and paste them in these boxes. So let's do it. So let's open this seq file first. As you will open the seq file, you can see the sequence. So let's copy the sequence. Now keep it into mind that this is a sequence which is extracted from the PDB file. And let's paste these sequences in the first box. In the second box, let's paste the full-length protein sequence. So this is a full-length protein sequence. So we are going to copy it and we are going to paste it here in a second box. After doing it, let's click on this submit button. And as you will click on this submit button, the job will be processed, and after a few moments the pairwise sequence alignment will come in front of you. So our pairwise sequence alignment is done. As you can see here in our pairwise sequence alignment, so you are going to observe the gaps at the two positions. This is the first position where there are the 3 gaps, and this is the second position where there are the 14 gaps. How we would have an idea that there are the 3 gaps and the 14 gaps? The two ways. Number one, you can count these dashes, and number two, you can count these gaps in the protein data bank file as we have done previously when we observed the missing amino acid residues into the protein data bank file. Now we have an idea that where are the gaps and how many gaps. Now by using this information, we will construct the alignment file. To construct the file, please use this alignment file. We have provided the template with this video tutorial. Please open it. When you will open it, then this file will be like this. Now to fill this file, first you need to open the SEQ file which is extracted by the script 1. When you will open this SEQ file, then copy its content and paste it here. After copying and pasting the content of SEQ file, now let's open the full-length protein sequence file which is downloaded from the protein data bank. And in this case, just copy the sequence. Don't copy anything else, just copy the sequence and paste it here between the colon and the steric sign. Now after pasting the full-length sequence in this file, now this is time to introduce the gap into the first sequence, the sequence which is extracted from the protein data bank file or the PDB file. Now where we will introduce the gaps? Let's have a look. Just go to the pairwise sequence alignment which we have just generated and copy the 4 amino acid residues before the gaps. In our case, the 4 amino acid residues are Y, H, L, and N. After copying this sequence, come into this file and let's find out this sequence. As you can see it here that this is Y, H, L, N. Now there are the 3 gaps after this sequence. So let's enter the 3 dashes. Now we have introduced the first 3 gaps. Now after introducing the first gap, this is time to introduce the second gap. Now where is the second gap and how many gaps we need to introduce? Let's move back to the pairwise sequence alignment. Here once again have a look at the last 4 amino acid residues before the gap. In this case, these amino acid residues are N, Y, I, T. You can copy them. Let's move back here and search this sequence here. As you can see it here, there is our sequence NYIT. There are 14 gaps after this. So let's press these dashes 14 times. Our alignment file is ready. Press Ctrl S and our alignment file is now saved. Now this is time to close this file and let's move back to the modeler. Now this is time to run the script 2. To run the script 2, once again, let's type mod 10.1 space script2.py and let's hit the enter button. Now this script may take some time to execute. Now basically what the script number 2 is doing, the script number 2 is going to fill the gaps. That is basically going to introduce the loops in the gap. As you can see it here that the script number 2 is successfully executed. Let's move to the repair 1 folder. When we will come to the repair 1 folder, you can see some extra files which are generated here. Among these extra files, these 3 files are important for you. These 3 files are basically the 3 different models which are built by the modeler for you. In these 3 models, the gaps are filled. Now which model is better? You can have an idea about the better model by looking at the log file of script number 2. So let's open the log file of the script number 2. If you will move down to the bottom, here you are going to find out the score, the mole PDF score. Smaller the mole PDF value, better will be the structure. So by looking at these mole PDF values, we are going to have an idea that this model is a better one. Now this is time to run the script number 3. What the script number 3 will do? The script number 3 will basically refine the loops which are generated by the script number 2 in our protein data bank file. But before running the script number 3, we need to have a small editing. So let's open the script number 3. This is a script number 3. In these two lines, you can see it here, the missing amino acid residues numbers are mentioned here. In our case, the missing amino acid residues are at the 2 position, one from 133 to 135, and the second one 217 to 230. And all of the missing amino acids belongs to the chain A. In your case, these numbers could be different. So before running the script number 3, please make unnecessary changes here. After making unnecessary changes, let's move to this black color window and run the script number 3. Running of script number 3 is just like the script number 1 and the 2. So type mod 10.1 script 3.py and hit the enter button. As you will hit the enter button, it may take some time. And after the few moments, the best predicted model will be refined for you. So as you can see it here, the script number 3 is successfully executed. And all of the gaps in the protein data bank file is now filled. So let's open the model. So we have selected this one. So let's open into the PyMol. So as you can see it here, that this is your model in which all of the spaces has been filled. And now you can use this model for the docking studies and the molecular dynamics simulations. Please let us remind you that if you have a protein structure file and there are the missing amino acid residues, and you are going to use that file for the docking or molecular dynamics simulation studies, then definitely you will end up with the error. So if you have a missing amino acid residues, then first choose the modeler, fill the gaps, and then go for the docking and the molecular dynamics simulation studies. Hopefully, this tutorial will be helpful for you.